Hey, welcome everybody. This is our April Twilight meeting via Zoom. It's going to take the place of any normal Twilight meeting we might have done in past years. It'll have to do for now. I have Joshua Bruckner from MDAR who's going to go first and talk about Spotted Lanternfly. Josh, I don't know your title at MDAR, but Joshua Bruckner is going to tell us about Spotted Lanternfly. Go ahead, Josh.
Yep. Thank you for that introduction, John. So as he said, I'm Joshua Bruckner from Mass Department of Agricultural Resources. My title is Forest Pest Outreach Coordinator, which means I'm in charge of education, outreach, and awareness of various invasive insects throughout the state, although we've primarily been focusing on spotted lanternfly because it is a growing and emerging pest of concern in the Northeast, especially in Pennsylvania. I'm sure if you're at all involved in pests or pest management or agriculture, then you have heard a lot about it. You can see what it looks like right here on the screen. We're displaying this little poster here, which shows the adult lanternfly at the top, and the bottom are the other life stages, including the eggs and the nymphs. So this poster can be downloaded at this link right here, massnrc.org/pests. I can provide the link in the chat box when I'm done with my segment. But lanternfly is a pest of concern because it is a sap sucker. It is closely related to things like aphids and stink bugs. It uses its proboscis to suck the sap out of plants. And it has about 70 different host plants that it goes after. Tree of Heaven is its preferred host. It is what we find it on the most. Although it does not need Tree of Heaven to complete its life cycle, and as a generalist, it'll go after a huge number of other plants, including things like grapes, hops, black walnut, maple, birch, willows, elm, a lot of other crops. We've found them on things like apples, cherries, cucumbers, pretty much everything. But grape is kind of the agricultural product that we would be most worried about, since both the immature nymphs and the adults seem to really favor feeding off grapevines throughout their life. So this photo here just kind of shows that this is from a Penn State extension. Kind of does a good job of showing what the egg masses look like, these kind of grayish little ovals, but they can be laid on basically any flat surface. And this is the time of year when eggs are still around. The eggs will hatch in late spring, kind of late May, early June, and then before the nymphs are active. So you still have time, especially in more northern states like Massachusetts, to check for signs of egg masses on basically any flat surface, not just host trees like Tree of Heaven or cherry or near grapevines. Check pretty much everything. So as I said, they are a sap sucker, and if you're looking for signs of damage, I mean, the insect itself is pretty vibrant, as you can see here. You know, when its wings are folded, even then, without their bright red underwing visibles, you can kind of see here. It has this distinct shape. You can kind of see the red wing coming through the mottled forewing. But if you're looking for damage, as a sap sucker, you're going to see, like, you know, these oozing wounds on the tree. You're going to see the sap flowing down the tree, and you're going to see a couple of things. One, there's going to be this buildup of honeydew, which is its waste product. It's a sticky, sweet substance. Kind of looks whitish when it accumulates in large numbers. And that honeydew is going to do a couple of things. One, it's going to promote the growth of a sooty mold. You can see the black stuff in the left picture on the bottom. That sooty mold is going to lead to plant death. It's going to suffocate, you know, grass or other plants that it grows on. The honeydew is sticky and kind of hard to get rid of. So if it's on like cars or sidewalks or the sides of buildings, that's going to be a huge nuisance. It will attract other nuisance insects such as wasps, ants, bees, hornets that are going to feed off that sticky sweet substance. There are some companies in Pennsylvania that have actually they make honey out of bees that feed off of the honeydew, eating all that digested sugar. And plants that get fed on in large numbers are going to see a couple different effects on it. Those plants are going to see reduced levels of photosynthesis. Fruits are going to produce less sugar. The plants have less hardiness and cold tolerance. In general, the lanternfly, with the exception of Tree of Heaven and grapevine, does not directly kill the plant, but is a huge stressor. So with other factors, it might kill the plant off. If it's feeding off of a sickened or weakened plant, it might kill it. but it's not going to on its own kill a healthy like maple tree or apple tree. Again, tree heaven and grapevine are kind of what they have the biggest impact on. That said, these are the signs to look for: the sap running down the tree, the honeydew and sooty mold buildup, and the insect itself, because the insect does tend to swarm in large numbers when there is a population of them. In Massachusetts, we have not found any living spotted lanternfly. Yeah, there's only been a couple of dead ones that have made their way into the state, but in states that have established populations such as Pennsylvania, New Jersey, Connecticut, New York, Virginia, if there's a large amount of them, this is what you're going to see. Most likely, they're going to be swarming in large numbers. And you'll notice they're not going after the fruit itself; they're going after the plant. In this case, the grapevine, or whatever else, other plant the fruit is growing on. But again, repeated feeding is going to the fruit's going to produce less sugar, so it won't taste as good. That fruit itself is going to get covered in the honeydew. You can kind of see on the leaf on the bottom part of this picture here, it kind of looks very wet and shiny. That's the honeydew, the sticky waste that is coating the leaf. So you can get sooty mold all over the fruit, which renders it unsellable. And as I said, grapevine and tree of heaven are the plants that we see most commonly end up dying from repeated feeding from spotted lanternfly. And in very severe cases, that can happen. This is from a vineyard in Pennsylvania. And some of them have seen up to 50 or 70% loss in fruit yield and in plants from repeated feeding of spotted lanternfly. You can see a lot of grape vines have been lost here from the spotted lanternfly. Some vineyards have gone out of business. So it's definitely very important if you're working in the agriculture industry, especially with grapes, to kind of monitor this. Check for signs of damage. Check to see if the lanternfly are on the plants at all, or on the kind of the perimeter of your property. Fences on any trees, especially host trees such as tree of heaven, maple, birch that are surrounding the property. Check for signs of them. You definitely, no matter what you find, whether it's an egg or signs of feeding or the adults or nymphs, would want to report it to the relevant state that you're in. Massachusetts, you would want to go to massnrc.org. I'm going to put that into the chat once I'm done talking so you can have that readily accessible. But just to show you what a really bad infestation looks like, this again is pretty obvious, but this is what it would look like. There's a lot of spotted lanternfly in the area. This is a video someone took in Pennsylvania. You can kind of see they just kind of swarm indiscriminately on whatever tree they're feeding on. This is probably a maple tree. Just climbing all over each other. The adults have strong enough proboscises to go through the bark of a tree. The nymphs are going to go after softer, more succulent plants and parts of the plant, like the leaves and the stems, or softer plants such as roses is a pretty common host of the immature insects. But as an adult, they're definitely going to favor woody plants and woody trees, again, grapevines and tree of heaven. So you definitely want to look for that. And we actually have the website right there. So you can see massnrc.org/pests. And at the top of the page, you can see there's that red link that says report a pest. That's where you'd go if you found a spotted lanternfly. You could let us know right there. Upload pictures that you find. Mass Pests is our Twitter account, which has news and information. And this website right here is where you can order free outreach materials, such as these ID cards. These have a link to our reporting form on it, as well as pictures. Pretty good thing to keep around on your person. So that's all I had. I'm just gonna upload those or put those links in the chat right now, and so you guys can have that. And are questions saved to the end, or should I just pass it on to whoever's next?
Go ahead, Liz.
I don't see any questions in the chat. Does anybody have a question quick for Josh, or shall we move on to our next speaker? Thank you, Josh. That was really helpful. We really appreciate your coming and talking with us.
Oh, you're welcome. I do want to comment on one thing I'm seeing in the chat. I'm seeing people talking about Bavaria bassiana, which is I just put the link to our fact sheet there, which has a lot of good links to files you can download and print, as well as our reporting form. Beauveria bassiana is a fungus that was looked at as a biocontrol that could target spotted lanternfly. It does go after spotted lanternfly, but I was just in a talk that was talking about this. In a large scale study, it doesn't seem to have a significant effect on a huge scale. So it's not recommended as a control as of yet, but it has been looked at as a biocontrol. As for pesticides, I can say this very quickly, they're susceptible to a lot of different things, and most studies have shown that dinotefurins are kind of the most effective against adults, but depending on the state you're in, there's gonna be different recommendations. Massachusetts, we're not at a pesticide stage yet because we don't have an established population. New Jersey does use pesticides, but I think they are moving towards limiting or banning dinoteffurin use throughout the state. It's gonna be different in Pennsylvania, so always check with what the local regulations are.
All right. Thanks, Joshua. I think we're going to move on just because we got a pretty full agenda, I think, Liz.
Yep.
So that's great because people will keep an eye out for it, and let us know, or MDAR know, if you think you see any. Thank you.
Yep. Thank you. You're welcome.
Okay. Next is Elsa. Elsa Pettit's our faculty grape person, and I don't know what she's going to talk about because I don't do grapes, but she's going to do it.
Yeah, so thank you, Josh, because I work with grapes, so I was kind of concerned about this. And I'm now a follower on your Twitter account, so to make sure. So I'm Elsa Petit, and this is my email address in case you want to write it down. But at the end of the talk, I will, if you are, interested in learning more, I also mention it all along the talk. So I'll be brief today, just a few updates. You might know, most of you might know me, but just so that you remember, I work at UMass and I teach viticulture and plant diseases class. And also my research is on grape microbiomes, and they could be useful for biocontrols. For example, I work on resistance to disease in hybrids, and I have more applied project with growers. As of this year, as Sonia retired, I have taken up a role, and trying to fill in that role is not easy, but so I'm participating in the Twilight meetings and the field days. And so you can follow me on Twitter, and it's free, of course. I'm saying this is free because there is a newsletter that is not free, but not really expensive either. And so if you wanted to go on and use those codes, it's pretty easy. I just put all of those codes each time there is a website or something that instead of typing the website, you can just scan the code with your camera on, with your phone. Most phones have that. So you would just point your phone to the code and not take the picture, of course, but just with your camera on, just point to that. So yeah, you can have access to the Twitter directly. So on Twitter, I just post things related to what's going on in viticulture in general, everywhere, nationally and internationally, and in the region as well. The newsletter, we work on that mostly every month during the dormant season, and every week to every 2 weeks during the growing season. So if you want to help us sustain the program, it's $10 yearly. It's not much, but it helps us fund the program. So if you wanted to subscribe to the newsletter, this is the barcode to access the documents to subscribe. And then this is in case you wanted to use not a check, but the bookstore. If you wanted to sign up for the newsletter through the bookstore, this is how this is the code you would scan. Okay. So that's just for the background info if you want more information all along outside of Twilight meetings. For updates, this is really what's important to know is which stage we are at with the grapes. And so now we are at least in Amherst, as I was looking at today, I was looking at Concord, we have not too far from where I live, and it's definitely full bud swell. Of course, that depends on where you are and the variety you're growing. So by now, because of we are at that stage, pruning should definitely be done. And you might have noticed if you've done it late, you have some bleeding. This is normal, but really now it should be done. Dormant spray can still happen, and you should stop that once you see green tissue just because of phytotoxicity. Some people push it a little bit far, but you might have phytotoxicity after you see green tissue. And by that, I mean, if you were to look at the different bud stages, the leaf basically would come up at some point. You know, when you get to bud break and leaf emergence, you would see those green leaves. Now we are really at bud swell, so you don't see really the green tissue yet. So it's still protected inside. So as far as disease and pests that we have to worry at bud swell, so this is bud swell. In terms of insects, we worry about cutworm and climbing worm now, and we can still control them. And for disease, we could make sure we don't have and control, and we don't have phomopsis, black rot, powdery mildew, and anthracnose. And I shouldn't say we don't have, because we all have it, but we kind of want to have that under control. So as far as what to do for dormant sprays and sanitation for diseases, you can see here you have a phomopsis. So it's a bleach wood with the little phomopsis, the fungus structure here. And so that's a sign of the disease. So if you see that, you have phomopsis, and it's very common. So you would see that. Another disease that is common to see now from last year is actually black rot, and you would see the mummified berries that still hang on the cluster. You don't want to keep that. So for dormant spray, if you wanted to start clean, let's say, it's not going to cover the whole season, but it's going to help reduce the disease actually to start with. So this is for phomopsis, powdery mildew, black rot, and anthracnose. And so what you want to do is really target the whole wood tissue, right? You want to cover the whole wood tissue, and you have different material that you might be or might not be familiar with. You have lime sulfur, sulfurics, coprofix, other copper products, sulfur, and stylate oil or any other oil. So as I say, you should not do that after you see green tissue because of phytotoxicity. We have toxicity of copper at cool temperature and sulfur at higher temperature. And so again, to start clean, you want to sanitize the vineyard, and so remove dead cuttings that could have phomopsis. So you might have done that during pruning, but you don't want them to hang in the floor of the vineyard. You also want to remove the clusters of the mummies that I showed you here on this picture. You don't want that because they have the fungus in those. So that's pretty easy, easy to say. It takes time, but start clean and remove those. In terms of insects, I mentioned grape flea beetle and climbing cutworm. And so for that, really, the flea beetles, they are fleas and they jump around like fleas do. And so typically you would scout for either the bud damage, so bud that look that they've been eaten, or the adult. So this is what they look like. And so if you have more than 2% bud damage, then it's advised to spray to avoid losing too much. The climbing cutworm, it's the same, except you would not really see the larvae. The larvae is feeding on the buds. And so basically you scout for the injured buds, and the same level of injury, 2%, you would have to spray. And if you see that you have problems with the bud being eaten, then you might scout for the larvae under weeds, in the ground of the vines, because they are not, you don't see them typically during the day, like in the top of the vine. So dates coming up tomorrow, and so you might have received that from another place, but tomorrow there is a webinar at 3 p.m. By Penn State and Cornell. And so basically for small wineries, so if you want to sign up, again, the same barcode is showing up. So you want to either take the address, write the address, or scan that, and it's free of charge, but you have to register. Another seminar that is coming up, I think Elizabeth Garofalo sent it from Yukon, Pick Your Own Residual. So All right, so that's it. And so I just put my email address again in the form of code. I had a lot of those. I like those scans. I think Rémi Pinheiro pointed that to me. So now I'm using them all over.
Thank you, Elsa. I realize, this is typically a tree fruit Twilight meeting, but some of the stuff that applies to grapes, sanitation, early fungicide spray for, sulfur or oil sprays help clean the orchard up, similar to they do with grapes.
Thank you.
I'll send you your grade at the end of the day. All right. Great. Thank you, Elsa. Perfect. All right. Jaime, you ready to roll?
Yes, I'm next. Thank you.
Very good. Go ahead.
Okay, very good. And good afternoon, everyone. Today, I will be speaking about two different things. One is a new insecticide, which I was discussing last year. Is Verdeprin. And this material, it was registered in late 2019, and it became available late in the summer in 2020. So perhaps not many people are familiar with this insecticide. So it's marketed by Summit Agro USA. You can buy it only from Helena under the formulation, which is 100 SL. And that means 100 grams of active ingredient per liter of the soluble liquid concentrate. That's the technical stuff. So Verdeprin is very similar to Altacor and to Exirel. They belong to the same class of insecticides, which is 28. That is the antranilic diamides. So when you apply this material, the insects are going to be affected by releasing calcium in the cells. So basically they will die very quickly. They will stop feeding. And this material is effective by ingestion, by feeding, but also by contact. Most diamide insecticides are translaminar. That means that the material goes under the epidermis of the leaf and is going to form a reservoir. There is some compounds, active ingredients in this class, which are more systemic. I am not 100% sure to what extent Verdeprin is systemic, but those systemic materials, I think, are basically because the leaves, the foliage, are going to absorb the active ingredient. And it provides long residual activity, and it's also a broad spectrum insecticide. So again, it belongs to the IRAC. IRAC is an acronym, and it means Insecticide Resistance Action Committee. They take care of the whole situation with the resistance. So they belong to the same chemical class as Altacor, and you can see the active ingredients at the bottom. It's just 5. Well, the label contains materials which are, or insects, which are affecting palm fruit. You can see some examples on the left. But actually, it's labeled for many insects. And what you can see in the asterisk, the 2 red asterisks, is just suppression for those pests. But it's also labeled for Persilla. And according to the label, you can treat the first generation nymphs using the high rate of 11 fluid ounces per acre. And according to the label, performance is enhanced when you use an effective adjuvant. Fluncurculio is also in the label. Last year in May, we did a comparison of Verdeprin versus Avant, and it was comparable. So it was good performance, but it was a small test. So according to the label, you can also need to go to the high rate, which is 8.2 to 11 ounces per acre. And the re-entry interval is only 4 hours for palm fruit, for stone fruit, and I believe for grapes. And the pre-harvest interval is 7 days for palm fruit and stone fruit. So these are the restrictions. Basically, do not spray Verdeprin more than 3 times per pest generation to minimize resistance problems. Do not use Verdeprin below the label rates, either alone or in tank mixtures. So make sure you are using the label rate, the lowest, but it has to be within the label rate. And you need to target the most susceptible stage of the pest. For example, in the case of the stink bugs, it has to be the nymphs. And one more restriction is that do not apply more than 33 ounces per acre per year. So as many insecticides, this verdeprine material is highly toxic to bees. So therefore you need to avoid treating when the trees are in bloom. But there is something interesting in the label. So when they talk about bee toxicity, they say that it doesn't have an extended residual toxicity. So I will explain what this means. So according to the label, it has to be applied excluding when the trees are in bloom. But this concept of residual toxicity is about residues on the foliage, and it's about the toxic effects on honeybees. So research that it was done in this area, they found that when you apply verdeprine to the foliage, then the residual effect is very short. So that means that there is not high or significant effect on honeybees after you spray. So because of this situation, the label says that this product has been determined to have a short residual effect. It's not against the pest. They're not talking about the pest. They're talking about honeybees. And it's called RT25 because it's going to kill 25% of the population. So basically what they're saying is that you cannot use this product when it's in bloom unless you applied a lower rate. You can see the rate, and the application has been made 2 hours prior to sunset or 8 hours prior to sunrise. So there is a way to spray this material if there is a need. So it's just one question. On which insect do you think we should focus for the comparison of verdeprine against a different material? For example, imidan or Avant. So do you think it should be tested against Persilla or plum curculio or apple maggot fly or internal Lepidoptera or a different pest? But I don't have the option, so you can type in your chat which other pests you are interested in knowing how effective this material is. There has been some work done by Washington State University on Persilla, and I think it shows that it's also effective at controlling the nymphs. That was Washington State, maybe different in the Northeast.
Jaime, while the poll finishes out, would you like to take a question?
Sure.
So the question is, what are the advantages of verdeprine over imidan?
Well, it's also broad spectrum material, but it's considered to be reduced risk. Basically, under proper applications, it's going to be less toxic to or less risk to beneficial insects. The other one, it will be that imidan also degrades very quickly or more quickly with rainfall. It's going to wash off. So my understanding is that verdeprine, because it's translaminar effect, is going to stay on the leaves for a little longer. But I can tell you for how long, after, not right now, because I don't remember exactly how many days or how much rainfall you need to get. So that's one of the, I would say 2 answers. It will be, one is reduced risk insecticide. That's the definition. And the second is that it's going to be less prone to washing off by rainfall.
Excellent, thank you.
Thank you. So here results, it seems that plum curculio followed by internal Lepidoptera and then Persilla are the ones who are more. I should be able to do some work this summer. I requested material, so it's coming soon to Belchertown. Thank you. So I think I will continue now with the second part. And it's about the brown marmorated stink bug. Maybe you're thinking it's too early to discuss this. Well, maybe not too early. I would like to show you some information collected last year. It's coming in a table. So this invasive species has become established in Massachusetts. Every single farm, it was 10 different farms that we monitored last year, it was present. So the table is here. I am not giving the name of the orchards, but I can tell you the deployment date. It was in late August and early September. you can see in red how many stink bugs we caught, the traps and sticky cards or the ghost trap. They were able to capture in just 4 or 5 or 6 weeks. It was not the whole season. It was just a few weeks in late summer. So at the bottom, you can see that there were more than 1,300 brown marmorated stink bugs in 10 orchards. There was one orchard that has high numbers of stink bugs in the farm, but you can see even low numbers is everywhere. So the way you can monitor for this pest, and there is published information, I will not discuss this in detail today, is clear sticky cards baited with the pheromone. But what I would like to discuss today is what is called the ghost trap, which there is no thresholds, there is no numbers that you can use to make a decision about when to spray. But I believe the ghost trap can be useful to kill stink bugs when it's close to harvest. So let me go to the next slide to explain this. So just a few weeks ago, we were notified that one proposal that was submitted for research, it was funded by the Northeastern IPM Center. So the title is in blue, and the goal is to be able to develop a IPM strategy that may allow growers, for example, pick your own in small farms, to kill as many as possible, to kill as many brown marmorates as possible from midsummer until harvest. So the support is for 2 years. So we're going to start working on this in the summer of this year. And what we are going to be doing is basically trap cropping in combination with the pheromone, in combination with the ghost trap. So trap cropping is, the definition is that you are going to use plants which are very attractive to the pest, more attractive than the cash crop. And there has been a good amount of research in different states, in Florida or in New Jersey, in other states, showing that for stink bugs, including the brown marmorated stink bug, you can use, for example, in the picture, you can see buckwheat. It's a cover crop, and it grows very fast. I think everyone knows about buckwheat, but in Florida, Dr. Russell Messick, he has been doing a lot of research in the past 20 years with native stink bugs. So he was testing sunflower, early blooming, early soybean. He was testing triticale. He was using sorghum. He was also using millet. And he found that for summer, sunflower and buckwheat are very useful, and you can add or complement sorghum and millet. So what we did in 2020 in Belchertown, it was a very small study, and the goal was to determine whether we can use the pheromone in combination with the trap cropping to be able to bring more brown marmorated stink bugs to those areas compared to the pheromone in the absence of trap cropping. So we compared sunflower, dwarf sunflower, doesn't grow very high, it's like maybe 2 feet in tall, sorghum, millet, and buckwheat. So we have an area in Belchertown where we had all these treatments, sunflower, buckwheat, millet, and sorghum with the pheromone. And then other plots where we have the same trap crops, but no pheromone. Then we have the pheromone alone, and we have a unbaited control. For this study, I was using sticky cards. So this picture shows you the plots on June 15, so they were growing. Then you can see on July 20th, and buckwheat was in bloom. The circle is the sticky card with the pheromone. And just very quickly, the results. I am combining all trap crop species because there was no difference. What you can see basically is that when you have a pheromone in combination with the trap crop plants, you can catch higher number of, the sticky cards can catch higher numbers of brown marmorated stink bugs compared to the pheromone in sticky cards in areas where there is no trap cropping. So using this information as a preliminary information, we submitted this proposal, so it's funded. So now what I would like to do, or what we're going to do, is an evaluation in 6 different farms plus Belchertown. So we already have 2 or 3 growers who are interested in this research. If you are interested, please let me know by chat or by email, because it's going to be a very simple comparison. We're going to be using sunflower. The plots are going to be around 100 feet long, 30 feet wide. So there is going to be a mix of sunflower and buckwheat. And this is because sunflower takes a long time for the plants to grow. So buckwheat in 4 weeks is blooming. So then we're going to have, for that reason, buckwheat in combination with sunflower. Then we're going to have a ghost trap. We are not going to spray insecticides to these blocks, to these plots, because the sunflower and the buckwheat also attract a lot of beneficial insects. So we don't want to kill them. We're going to be using a small ghost trap with the pheromone. And this ghost trap is also going to be tested in the absence of trap cropping. You can see the grassy area. So same area or similar area in dimensions. So then once a week, once the buckwheat or the sunflower are growing, one person is going to go to each farm, and this person is going to count how many brown marmorated and other stink bug species are found on the ghost trap dead in these 2 plots. And this person, well, the person is also going to spend some time checking on the plants to see if there is other stink bugs or present on the plants which they haven't been killed by the ghost trap. So it's a very simple comparison. It's going to be done every week for maybe 15 weeks, maybe 14 weeks. So from June to September, perhaps into October, it depends. So that's all what I wanted to say. And if you have any questions, I don't know if there is time. Thank you.
Thanks, Jaime. That was great. We did have we had actually 2 questions come in. One of them you've already answered. And the other was about the vertiprin, and it was, what is a good adjuvant for vertiprin? What is the price? This is all in the same question. What is the price? And
well, the answer to these 2 questions is I don't know. I can find out about the adjuvant, which one is the best option. And that's the honest answer to the first question. When it comes to price, I also don't know. But what I can tell you is that when I was checking for a similar material, which is labeled for vegetables, I think it was not this material, but the other material. It was $400 per gallon. So I'm not saying that it's the same price, but I can also find out and maybe have information in the healthy fruit, because I just didn't have a chance to find out about the price. Plus, the first answer was about the adjuvant. I can tell you, not at this moment, what are the options for adjuvants. Thank you.
Given that the residual toxicity to bees is low, what is the residual toxicity to pests?
I had the same question when I read the label, because if they're testing this against honeybees and they find low residual effect, I think what is happening is that they're assuming that the pests are going to be chewing the foliage, for example, and that is something that the bees will never do. So I guess with the honeybees, it's mostly landing on the foliage and being in contact, as opposed to feeding, because that will be the best route of control for the insects, basically active feeding on the foliage. I think that would be the difference. Plus I want to say also that even though it's not explained, it's also based on size. So you have small insects versus the size of a honeybee. That may also count into the toxicity. Less toxic because they're bigger, so they can take a little more material. I'm just mentioning this the way you understand it, but it's not explained in the label.
That's great. I think that covers everything. Thank you so much, Jaime.
Thank you.
How about I go now?
Dwayne, why don't you hold off for 5 minutes, and before I forget, we'll let Heather give
Thanks,
a little
John.
talk here, okay?
All right.
Stick with the
Okay.
insects.
Stick with the insects, right. So yeah, I just want to talk, of course, I always talk about winter moth, and winter moth populations are way down. They started hatching earlier than normal this year, which we would expect since it's been warmer than normal, but started seeing them hatch around the end of March. But I really don't expect them to be a problem. Yeah, they start hatching right around green tip usually, or when blueberries look about like this and the caterpillars get into the buds and destroy the flower buds. but I really don't think we're going to have a problem. The time to check your buds is when the apples are at like tight cluster, or blueberries at tight cluster, and you really just need to pull apart the buds a bit and look for damage. Let me go to the next one. So you don't often find the little caterpillars in there, but you'll often see the frass. So here are some little caterpillars. This is at a tight cluster stage in apple, and blueberries are a little harder to check, but you can find the black frass that indicates that winter moth have been in there. So we don't have a good threshold, but in Nova Scotia, where they've had these for decades and decades, they're saying if you're finding 1 or 2 damaged clusters out of 20 clusters, it would indicate a time to spray. And in Nova Scotia, they're still treating like 9 out of 10 years for winter moth. I don't see that happening in Rhode Island or southeastern Massachusetts. But it's worth going out and looking, especially a tight cluster, if you see any damage. All right, let me just, and then they're easy to kill. You can use just about any insecticide that you'd be using that time of year, and BT works well controlling them as well, since they're caterpillars. We do still have green pug moth, which are very similar, do similar kind of damage. They come out earlier than the winter moth even, and will damage flowers like this, eating out the flower parts. The tiny larvae look identical, so if you see them, you don't know which you have. Doesn't really matter. But when the caterpillars get bigger, you've got green pug on the bottom and winter moth on the top. I wanted to mention rosy apple aphid, because I was in an orchard last week and saw rosy apple aphid, which I didn't even realize it was rosy apple aphid. So you're probably familiar with this. You see that get the curled leaves and the pygmy fruit, though. I'm not sure pygmy fruit is politically correct to say. I'm not sure about that. But anyway, you get those tiny fruit that stay. But the way that rosy apple aphids look right now is they're kind of dark green. They will be getting some of this white powdery cast to them, but really when they first hatch and the aphids are small, they really look quite a bit like green apple aphid. So I was just sort of surprised. I was in this orchard and I saw a lot of rosy apple aphid, which at first I thought were just green aphids, but on closer examination, they are actually rosy apple aphid.
Are they actually that big now, or because they overwinter as eggs, right?
They overwinter as eggs, but they start hatching at like silvertip. And so there were some
they grow pretty fast.
They grow pretty fast, and we're still just in the first generation, but the adult females just pump out live young. They don't need to wait. They don't need to mate. So I don't think I was seeing some quite this big, but they were pretty big. But it was difficult to tell between them and the green apple aphid. They do have these dark corticals here. Their little tailpipes are quite dark, and there was a little bit of this white, this blush on them, the powdery that they get later, because we're really more used to seeing them looking like this, very rosy colored or purple colored. So that's usually the way we see them.
Yeah, and then it's too late.
Then it's too late. I'm finding a little bit of conflicting information. It seems like, well, in the New England Tree Fruit Guide, it's saying that a dormant oil at green tip to half-inch green will really do a good job suppressing rosy apple aphid. So that's a good thing. But and then when you get later in the season, you need really kind of serious insecticides to clean up rosy apple aphid. Most of these in the list on the New England Fruit Guide are restricted use pesticides, or they're really expensive also. So, getting on an early dormant, it might be a little bit late this year, but putting on a dormant oil spray can help suppress rosy apple aphid. One other thing, we think of rosy apple aphids being more of a problem during cool, wet springs. Well, this is not a cool, wet spring. But I saw so many at this one orchard that I was really kind of alarmed. And the cool, wet spring is just supposed to suppress predators. Well, I don't usually find predators around rosy apple aphids, at least not early in the season. So anyway, it's worth going out and looking for them. I think that's it. Fast enough, John?
Yeah, too fast maybe, but that's okay. Any questions for Heather? I'm finding more rosy apple aphids around. I don't know if they're building up. we seem to miss the treatment window, and then you've got them and you really can't do much about them. I mean, it's not like they're orchard wide or anything, but where they occur, they're just nasty.
Right, and so actually, yeah, they're supposed to be worse on Cortland and Ida Red and Golden Delicious. But I was in a
Delicious.
cider orchard, and there was, I'm not sure what the varieties were, but there were these cider trees were really infested with them.
Okay, and it just seems like winter moth has become less of an issue overall.
Yeah, it's worth scouting for it, you know, when you're at tight cluster or so, but.
Just, okay, and if, all right.
And if you, yeah, if
Okay.
you're finding like 5 to 10% damage, you'll want to spray.
Dwayne, you all set?
Okay, well, I've been asked to talk about use of apogee, particularly the pink application timing. And we're getting close to that, so you can start to think about it. Why apply peroxide ion calcium at pink? I'm going to use peroxide ion calcium because both apogee and kudos are the two product names. And so rather than just select one or the other, I'll use that name. There are two reasons to do that. One, improve growth control, and then for a different approach on fireblight control. So first of all, I'm going to be talking about growth control. If you want to get the most out of peroxide ion calcium, you really need to apply it early. And about the earliest you can do it is the pink stage. If you think you're going to get enhanced growth control from adding, applying a higher rate of peroxide ion calcium, it just doesn't work. And I'll show that shortly. But the application here are the application particulars. It's very similar. It's just review, but the apogee label is the same for the pink application as the others. Kudos restricts the amount you can apply to 6 ounces per 100 gallons. You have to use a water conditioner or an equal amount of ammonium sulfate, and follow-up applications are appropriate if you want to maintain growth control. And I define pink as when there is sufficient leaf area to absorb the peroxide ion calcium. So there's nothing magical about the pink stage, but you have to have enough leaf area in order to absorb the compound. Now, this figure is really pretty important. What it shows is the effective concentration or the amount of peroxide ion calcium applied on growth control. And there's a series here, 30, 60, 90 parts per million, and the 270 is off label, so I will not talk about that. But if you look, I guess yeah, here's my pointer. You can see these 3 lines, they're virtually indistinguishable. So regardless what concentration you put on, you're going to get more or less the same amount of growth control. The difference in the concentrations is how long it will last. So you're not going to get any more out of growth control by jacking up the amount you put on. And the only way you're going to do that is by putting it on early. Now, I'm not going to show you any data. I've got 6 slides here. What I'm going to do is just show you the treated trees. The first 3, I'll show you at about harvest time, and then the last 3 I'll show you at dormant time. Here's a control Cortland. You can see it's rather vigorous, and there's a lot of shade in that tree. Here is a Cortland tree that was treated with peroxide ion calcium at pink, and you can see that it certainly did open up the tree. It reduced the terminal growth quite considerably. Now, one thing that I've noticed in looking at these trees, not only does it reduce the terminal growth, but it also reduces the lateral growth. So you can see all those holes in there in the tree where light penetrates. You can see that there's very little shade there. Now, before I was never, when we applied peroxidine and calcium at petal fall, I was never able to see an effect of this compound on red color development. It's not until I applied a pink could I show an effect of red color. And you can see why, because it opened up the tree and exposed a lot more of the fruit. And then here's the standard application that we use for years. You can see that there is growth control, but it's still, there's an awful lot of shade in there. And we didn't get the growth reduction that I would certainly like to see. Now, here is the same trees in dormant. And you can see this is the control. There's a lot of growth in there, a lot of lateral shoots, and the terminals are quite long. Here's the apogee. This happened to be apogee applied at pink. You can see that the terminals are much shorter. And if you look at this shoot, you can see those laterals are really short. So we've really reduced the amount of vegetative growth in this tree. And then here's the standard timing petal fall. You can see that there is reduction in growth, but it's not nearly as impressive as the time of pink application. Okay, so that's all I'm going to say about the pink application of peroxidine and calcium. I think that it's if you want to really get extra growth control out of peroxidine and calcium, that's the time to apply it at pink. Peroxidine and calcium also is useful to reduce fire blight. It helps control early blight. It has no direct effect on the fire blight bacteria itself, but it slows the shoot growth, thus thickening the cells, and it more or less impedes, provides a physical impediment to invasion. Pink application comes at the expense of inhibition of vegetative growth, and I'll mention this a little bit later. If you want to put these on young trees, you have a block of that's highly susceptible to fire blight. It could be cause a certain amount of problems. So you're going to have to modify your approach there. Now, having said that, I'm going to present Plan B, which was presented to me by Dan Cooley. The label modifications of these were made at pink for fire blight. Generally involved the use of 6 ounces per 100 gallons. And that occurred for 2 or 3 years. The application of petal fall at more traditional time appears to work very well without as much inhibition. So lower rates of production of peroxidine and calcium will reduce shoot growth, but you can have a resumption of growth earlier. Thus, you won't have the inhibition. So if you want to get inhibition early, you can not have follow-up applications and just let the tree grow. Now, here's something that Dan Cooley shared with me, and he got this from George Sundin, who's a pathologist at Michigan State. And this is one that Dan favors. 2 ounces of peroxidine and calcium plus 1 ounce of Actigard applied at petal fall of the king flower. Now, this is a little bit earlier than petal fall per se. It's the petal fall of the king flower. Now, how many applications you make will depend upon rainfall or perceived fire blight. Now, 2 ounces is not a great deal. So you'll start to have some regrowth after perhaps 2.5 or 3 weeks. And so reapplication will have to be made if you need to reinforce the inhibition or you want to have growth control. Now, Carrot Cox in New York has had good success by applying 6 ounces per 100 gallon. And he also had improved control with addition of Actigard. So that combination appears to be quite good. Now, I think I'd just like to mention a little bit on young trees because, of course, we're trying to we're trying to get these young trees to fill the allotted space, and applying a growth retardant seems to be quite counterproductive. One approach might be to apply peroxidine calcium at pink at a low rate, and no additional sprays may be applied. And so you can get early inhibition, but you'll let the tree grow, and hopefully it will catch up a little bit. Alternatively, use a lower rate of peroxidine calcium as suggested by Dan. Trees would then be allowed to resume growth partway through the year. Some total growth will be lost, but blocks containing susceptible varieties would be protected early. So that's a choice that I think you'd have to make. So in conclusion, application of peroxidine calcium offers growers the opportunity, the greatest amount of growth reduction and you can get up to 25% more reduction in growth. So it really enhances the effect of peroxidine calcium. Application of peroxidine calcium at pink offers growers much earlier growth control of shoot blight too, if that seems to be appropriate. A good compromise between growth reduction and fire blight is to apply peroxidine calcium at 2 ounces per 100 gallons plus 1 ounce of Actigard per acre, as Dan suggested. So that's it. John, I told you I'd do it fast, and there it is.
Seems to me that it's just a no-brainer. Go out and apply it at pink if you're going to use it at all.
I'm not as blunt as you, John.
Well, like
Well,
I said,
sometimes you don't want to have you don't want to hit the tree over the head, but in particularly younger trees where you may want to have you may want to have some fire blight control, but still you don't want to shut them down. And you really can shut them down. I know in Belchertown we have had after 2 years we've really changed the shapes of those trees, and so what we do is just wait a little bit later to put it on if we want to get a little bit more growth. So
yeah, I think some key points is you have to decide what your major objective of apogee use is. And I don't like it when people say fire blight control, because it really isn't control, it's suppression perhaps.
You're right.
You would still want to spray streptomycin, for example, at bloom if the conditions warrant it. But anyways, all right.
Yes.
Okay, thank you, Dwayne.
So I will go quickly. I think I have 3 minutes, you said?
You got 10.
I want to just say a few quick words about some new materials. Really, the news is about 2 new fungicides this year. To sort of set the stage, just a quick review. We have multi-site broad spectrum materials that we've had for ages, the Captan and the EBDCs, various kinds of mancozeb formulations. And then we have a whole bunch of single-site systemic materials. We have the FRAC 3s, the DMIs, and these contain they sort of are in 2 generations. There's a bunch of older DMIs that are gradually losing their effectiveness. In some places, they're completely gone, like Michigan and some parts of New York. But here seem to be holding on. But the very best stuff, Inspire Super, is still quite effective. And then there's a new material that's come out this year called Sevia, and I'll say more about that. The FRAC 7s are a big group of the newest chemistry, SDHIs, and there's my whole list of them. We have Aprovia, Fontellus, Miravis, Circadus. Where do they get the names? I'm not sure. Anyway, there's a new one this year, Excalia. And then the older materials tend to be premixes in this group. There's Luna Tranquility and Maravon. Then frac 11s, these are relatively old, the QOIs. Some places there's some resistance issues. Again, I think we're still doing okay. Flint Extra, Sovereign, and these sometimes show up in the premixes as well. Then there's a couple of analynyl pyrimidines, frac 9s, Vanguard and Scala. And then the oldie but goodie, in a really kind of strange and unique group, the frac U12 Sillet. So we've got these single sites and multi-sites. The new ones that were there in red are this Sevia. Now, it's a DMI, and my question was, who needs another DMI? They're on the way out. But this is new chemistry, according to BASF, and I was kind of cynical, but I talked to a number of my colleagues who've been doing tests around the country, and they say, yeah, this is good stuff. And I said, show me the meat, George, and he did. So that graph shows you, if you look at the performance in his blocks, which are definitely have resistance to DMIs, and you can see that with the results of Top Guard and Rally, compared to a couple of different Sevia rates. It does quite nicely. The label rate is 3 to 5 ounces, and I would really stress using, even though it's more expensive, the 5 fluid ounces, because it will not only give you better control, but again, it's part of trying to maintain the efficacy of this material as long as we can. And also put it with one of our protectants, Captan or Mancozeb. Efficacy not just against scab, but also against rust, black rot, sooty blotch, and fly speck. And so this is a useful material even in summer diseases, but it is limited to 3 applications per season. If you want to use, it might be very useful at the end of the season. I haven't really thought that one through or gotten an opinion one way or the other, but it has essentially 0 days to harvest. And so it might be useful at that time. I just had this other little graphic down in the corner to show you the scab efficacy of the older materials versus Inspire Super. And so you'd put Sevia right up there at the top in terms of scab. Interestingly, if you're looking at powdery mildew, just as an aside, they seem to work exactly backwards. So it's kind of a weird thing. Here's the other really new one. It's a new SDHI, a frac 7. I like it. It's not pre-mixed. It used to drive me nuts that the companies were pre-mixing their stuff. But now we've got a total of 5 straight frac 7 fungicides. As with the other SDHIs, it's very good on scab and powdery mildew, okay on rusts. In Sundon's tests, he feels that Excalia, this new one, and Miravis are the best. They're just a notch better than the other SDHIs that are out there. I think, probably in normal orchards, it's 6 of one, half dozen of the other, but obviously good chemistry, good material. Another quickie reminder here, this is to say that we're not really quite yet in the high risk zone in terms of apple scab. That will start in pink and then go through to petal fall. And this comes from another piece of information I've stolen from George. Anyway, he's done a nice job of listing why that is. For one thing, the spore load is heavier as we get later on into bloom. Warmer weather, shorter infection periods, increased growth means that it's harder to keep fungicide cover on, things like that. The whole potential for infection during that period is higher. Well, that means that up until that time, the infection is lower. And I recommend that we go ahead and use something like copper. We're beyond this, but then at this point in the year, up and through and into tight cluster, I think we want to look at our protectants, the broad spectrums, Captan, and the EBDCs. And then for systemics, and systemics are a good idea, I would combine them with either Captan or Mancozeb. But you could take Vanguard in one of those or Scala in one of those and put them on before the rain comes. Psyllitt is also still working pretty well for a number of people, and you could combine that. So those sorts of combinations getting us through into the high risk zone, and it's there that we want to look at using our most potent fungicides, the SDHIs, and DMIs, and QOIs. Again, a tank mix with a protectant. One final thing I'm going to mention here, that there's a new biological. You can see on the label, it's got an OMRI approval, so it can be used in organic agriculture. It stimulates resistance the same way that Actigard does. It's labeled for scab and other fungal diseases, but I think really it's going to be most useful as an alternative to Actigard, because that's what it does. I think the efficacy against the scab in particular is up and down depending on what tests you look at, and overall it's not as effective by a bit as some of the more standard materials.
A couple, I don't know that Excalia is registered yet in Massachusetts, and can only be used through pedal fall.
Mm-hmm.
Just a couple of notes. All right.
We did get a question come through about Lifeguard, and can you use it for fireblight?
The answer is, Peter, you can use it for other things besides fireblight, but it just is not, well, you might get lucky and you might not. It depends on the conditions that you put it on. It's one of these bacterial biologicals, and they tend to be uneven in terms of their control.
Suppression at best.
Mm-hmm. Somebody's asked, or Ben's asked, if it's too cold to worry about brown rot on stone fruit in full bloom this week. Good question, Ben. I would say brown rot is always enough of a concern that I wouldn't write it off completely. I'd be a little careful about putting on anything very, very oily, very harsh in cold weather. But I'd put something on if it rains like they say it's going to.
And
Okay.
is that only if you've got open flowers, or you think it should just go on anyway for the brown rot? Would you say?
I think generally, I wouldn't worry so much at this point about buds. I think if you start to get open flowers, then the risk goes way up, and you should do something.
Liz, you got something to present in 5 minutes or less?
I will do my best to zip through it quickly.
I'll give you 10.
Everybody's ready to get going. So I do want to take a minute or two to just talk about the fact that we are, depending on which source you look at, experiencing a drought in the state of Massachusetts. Our state's Department of Energy and Environmental Affairs has declared a level 1 drought for most of the state, level 2 for the southeastern part. The islands appear to not have any drought going on. What this means for us in terms of our tree fruit, of course, that we're going to have increased issues with pests that affect stressed out trees, right? So we're going to have potentially black stem borer issues if you've got younger trees that have gone in and they're drought stressed if you're not irrigating. Deer are going to be getting thirstier, and other vertebrates will become an issue. And of course, gypsy moth typically is managed with there's a fungus and a virus that are just sort of environmentally there. But when we don't get a lot of moisture, those organisms don't have an opportunity to reproduce. And so the larvae can go on and do their own thing, and they tend to balloon into orchards and into blueberries, which we had an issue with a few years ago. So it's a thing to just keep an eye on. I've got an egg mass that I'm monitoring out in the backyard, so I'll keep you posted as best as I can. But if you start to see them come into your orchard, the gypsy moths or any other little caterpillars of that nature, you really want to get on them before they get big, and BT is a good way to manage that. Of course, drought also really affects apple scab. And so as Dan was saying earlier, we're in that early period where we're not quite at our peak risk yet. And we're not actually seeing an awful lot of spore right now. In fact, I didn't actually see any ejected this week. Last week I did have some. This week I didn't have any ejected. And so what happens when it's dry, the development of the fungus itself actually slows and can stop. And so what that means is that we can tend to have a more protracted period. So right now we might not have a lot of pressure, but that doesn't mean we're not going to get it going down the road. Just so a kind of a sense of where I'm at right now with development is, so I had green tip here in Greenfield, which is where I'm doing my spore work right now, on the 31st of March. And just about yesterday I was at kind of early tight cluster. So in the amount of time it took us to go from healthy fruit being done this afternoon and going out to you to now, the actual forecast that we were seeing in RimPro changed. The change was that it actually decreased in severity. The infection event decreased in severity. So before, we were looking at an event that was going to start around the 15th and reach this medium infection risk 100 level here by the 17th. We're now not going to touch that. So again, just to back up what Dan was saying, we're still in that early period where we're not at really high scab risk yet. And in case you're not as familiar with RimPro as some of the rest of us are, I look at this a lot. We've got a couple of different things going on with this graph. Down here at the bottom, where you see the blue, we have dark blue is rain. So one of the things that has changed actually is the forecast. Previously, NOAA was saying we were at about an 80% chance of rain on Thursday. We are now, thankfully, at 100% chance of rain. The lighter blue here down on the bottom is leaf wetness. So as you can see, you've got that dark blue and light blue all put together there. That's the criteria that we need for an infection event. The temperatures aren't going to be really high. So again, that just contributes to the lessening of the severity of the event. Up above those blue lines, you see the red. The dark red is the immature ascospore bank. The bright red is those that have been estimated to mature. And then at the top, the big part of the graph here is where you actually see your infection event predicted. And that red line that you see slightly sloping up there, that's the RIM, the relative infection measure. The higher that line goes, the bigger the event. The other items are this dark orange here, that's that primary stroma. The spore has already penetrated into the leaf. And the white is the germinating spores themselves. That's really helpful if you have a tight window for application and you're looking to get something potentially in that immediately after that event has started and get a little bit of kickback going on. Powdery mildew is also a thing that we're thinking about right now. Powdery mildew overwinters in infected leaf buds. The mycelium are in under those bud scales. They come out a little bit later. They tend to come out a little bit later than other buds. And so what happens is that those later buds that are infected pop open, create inoculum, and then there's already green tissue to get infected present. So some of the more susceptible varieties are Cortland, Gala, Gingergold, Ida Red. If you had powdery mildew last year, you're going to want to keep an eye on it this year. So the thing with powdery mildew is it doesn't need standing water or free water on the leaf tissue. In fact, it can't actually germinate in that free water. It needs high relative humidity. Thankfully, right now our temperatures are going to be down low enough that it's not really conducive to powdery mildew development. But if you had an issue last year, tight cluster is about when we start to manage for it. And there's a number of different things that you can use for it. Dan mentioned Sevia. That can be used. It's in the guide is listed as highly effective. Indar Rally, also Flint Sovereign, the FRAC 3, the FRAC 11. There are organic options as well. It's a little late for oil at this point, but depending on where you are, of course, but a good dormant oil can suppress that as well. And you know I can't do a Twilight meeting at this time of year without talking about Perstilla. So the first adults we saw this year, or the first adults I saw this year, were March 11th in Belchertown. And we did see, or I did see eggs this week as well. So this down here on the bottom right is a kind of a really blown up picture of what the eggs look like. And I don't find that to be super useful in terms of like practical identification in the field. So I'm going to try to give you a sense of exactly how tiny these eggs are. If you're out scouting for them, you really do want a hand lens. I don't know, I can see this, but I don't know if you can. The arrows, those teal arrows, are pointing to a thin line right at the base of where the bud scale was, or the last year's bud growth. And you can see 5 little bitty orangey yellow dots. That's the eggs. So if you see those, you have the eggs. Now at this point, you've probably already gotten your oil onto your pears and you're protected for now. But these eggs, eventually, anything that went on, anything that was laid after that oil went on, you're gonna have to worry about dealing with those nymphs.
I just think you missed an important point talking about mildew, that if you're just spraying Captan and Mancozeb fungicides for scab pre-bloom or into petal fall, those are not going to get mildew. They're not effective on mildew. So if you have a mildew problem, you need to work one of those other frac groups in, not an M. A single site, not a multi-site, Dan.
How about Topsin M against powdery mildew?
I don't know.
I don't believe it's in the guide, but that's a good question, Heather.
Heather, why do you ask?
I've always thought Topsin was pretty much just a scab product.
No, it's good against summer disease. I asked because there's a lot of growers that try and just have a few pesticides and they'll use that in the summer for summer diseases mixed in with Captan. And so if it could work against powdery mildew, that would be one cheaper fungicide they could buy.
I think Liz can correct me, but I think the horse is out of the barn if you're trying to deal with mildew in the summer. John's absolutely right. I think you have to take out the heavy artillery that we use for scab and make sure that it also has mildew efficacy at the same time, from starting right from pink on through.
But you know, I mean, that does match up fairly well with your accelerated phase there. You know, your shaded area starting at about pink or a little bit later, when you suggested start using some of those other fungicides with kickback.
Right.
But all right, Dan, just to save myself, I was saying to use it early, to use the Topsin M, say at pink or at petal fall. Try putting it in early I was just wondering if it would work against protecting against powdery mildew.
I don't know off the top of my head, because it's gotten to be, if I think of it for anything, I think of it for sooty blotch fly speck.
Okay, thanks.
Well, it's apparently labeled for powdery mildew, so. All right, are we done? I'm done.
We have finished on time. Are there any questions?
Questions, comments?
Mo has said that a low rate of sulfur while it's cool, temperature-wise.
For powdery mildew?
Presumably.
Yes, sulfur is fairly good on powdery mildew. Is the question, is it good or is it okay to put it on while it's cool?
I think he's saying that it's a good material to use for powdery mildew while the temperatures are low.
Makes sense to me.
All right.
Okay, well, everybody, thank you for coming. I'm going to wrap it up if we're done, and thank you. Talk to you later. Good luck.